Hi everyone, my name is Teddy Starcheva. I'm a master's student at DePaul University, and I will be presenting on some of my thesis work, which looks at the effects of experimental warming and elevated CO2 on black spruce cone and seed characteristics. So to start off with a little bit of background, warming and elevated carbon dioxide have been shown to have individual effects on tree reproduction. So warming can either affect the magnitude of reproduction, or it can influence soil mineralization, its subsequent nutrient uptake, which can have implications for seed quality and seed viability. CO2 has been shown to have a fertilizing effect on reproduction, so it has been shown to increase reproduction with no negative consequences on seed viability. The gap in knowledge, however, is how these two environmental drivers of climate change together interactively influence cone characteristics and seed viability. So that's sort of what we're looking at, is how do temperature and CO2 interactively influence cone characteristics and seed viability in black spruce, because this can have potential consequences for seed viability, forest regeneration, and forest composition in the future as climate change continues. So the Spruce and Peatland Response Under Changing Environments, or SPRUCE experiment, is located in northern Minnesota. It's at the southern edge of the boreal forest, so it makes it an area that's very susceptible to the changes or to climate change. It looks at both temperature and carbon dioxide and its effects on boreal peatland ecosystem dynamics. So in terms of experimental design, Spruce utilizes these open top chamber designs. Each chamber, this is a picture of a chamber, each chamber is 12 meters wide by 8 meters tall, so they house whole trees in there. And there are 5 temperature treatments ranging from +0 degrees Celsius above ambient to +9 degrees Celsius above ambient in increments of 2.25 degrees. And each of those temperature treatments is paired with either ambient or elevated CO2 for a total of 10 treatments, which you can see here in this aerial view of the design. This is just what it looks like inside of the chambers. So it looks at whole ecosystems, particularly black spruce and eastern tamarack. I'll be focusing on black spruce today, but there are also some shrub and moss communities in there as well. So in terms of data collection, we collected cones in one of 2 methods. So for the first method, we did in late August to early September of last year. We obtained 300 new cones from 46 trees using what we like to call this ladder hook grabber method, whereby we kind of stand on a ladder and pluck cones from the top of the tree using a snake grabber. And you can see me doing that here. We collected, I think I said, 300 new cones from 46 of the trees, and then individually bagged them and labeled them. And then in October 2021, there was some cuttings at spruce. Some of the trees were growing out of the chamber, so they were just topped to contain them. And we obtained an additional 218 new cones from 27 more trees for a total of 518 cones from 73 of, I think it was like the 97 reproducing trees across all the treatments. We did also get 6 old cones from the warmest treatment with ambient CO2. Reproduction there has been shut down since 2018 due to warming, so we kind of just wanted to have something from that treatment to analyze. We then did some cone measurements and seed extraction. So we measured cone size, so the length of the cone, the number of scales, as well as the number of seeds, the seed mass, the cone mass, and then the total and mean seed mass for each cone. And we also conducted some germination trials on the seeds that we obtained from all the treatments. In terms of analyses, we actually saw some nonlinear responses to the treatments. So we used generalized additive mixed effects models. We built 4 candidate models that included a full model with temperature, CO2, and the interaction, a temperature-only model, a CO2-only model, and a null model. And individual trees were also included as a random effect in all the models because we collected cones from multiple cones from the same tree. We also found no significant correlations with tree size. So we didn't include tree size in models. And we used AIC to select the best model for each response variable. So in terms of analyses, for dry cone mass, under ambient CO2, we kind of see this curvilinear response, whereby with warming, cone mass starts to increase and kind of peaks around the +2.25 and +4.5 treatments, and then decreases at +6.75 and +9 above ambient. Here for the +9 ambient treatment, we've included zeros for cone mass for 2021, since those trees didn't reproduce. So it kind of made sense to include zeros since they didn't allocate any resources to cone mass. And these cones right here are the cones from 2018 from that treatment. You can tell they're kind of similar in cone mass to the cones from the 6.75 treatment. Under elevated CO2, we see this very interesting curve. So with warming, cone mass starts to increase and then decreases, with it being the lowest in the 6.75 treatment. And then the weird thing that happens is it increases again at +9. And you'll see this with the rest of the analyses too. These two treatments are sort of on opposite ends of the spectrum, where 6.75 is typically the lowest and +9 under elevated CO2 is the highest. And we think that's sort of because maybe at this 6.75 treatment, the trees are experiencing some sort of heat stress, and that's overcome at +9 due to that additional CO2. In terms of cone length, we see a similar response as cone mass. So with warming, cone length starts to increase, peaks at sort of 4.5, and then decreases again. With additional warming, we didn't include zeros for this measurement or for the rest of them, just because it didn't make sense to have a cone length of 0. But you can tell again, the cones from +9 ambient in 2018 are similar to those in +6.75. And then again, under elevated CO2, we see this curve where cone length starts to increase with warming and then decreases, with these two warmest treatments being again on the opposite ends of the spectrum. So for the number of scales per cone, this is just a picture of a black spruce scale. You can tell that at the very bottom of the scale, there is room for only 2 seeds. So each scale can have up to 2 seeds and no more. And because some of these cones were collected at a later date, it's possible that some of them opened and released seeds. So we just decided to run the analyses on the number of scales because we might be missing some seeds. It might not give us completely accurate results. So for the number of scales per cone, the best model here was actually the temperature only model. And you can tell that with warming or some moderate warming, the number of scales and presumably the number of seeds doesn't really change much until you get to that 6.75 treatment, where the number of scales is significantly lower than the other previous treatments, and then increases again at +9 degrees Celsius, with again those cones from 2018 being similar to the number of scales to those in the +6.75 treatments. In terms of the total seed mass per cone, so cone mass was increasing, number of scales and the cone length was also increasing or changing, but in terms of total seed mass, it is not being affected by the treatments. So temperature, CO2, or their interaction don't have an effect on total seed mass. There's a lot of variation in the data, but none of that is due to the treatments or the interaction. The same is true for mean seed mass per cone. So again, there's a lot of variation in the data, but it's not due to temperature, CO2, or their interaction. And that's kind of interesting because seed mass is related to germination success, right? So seeds with a greater mass are expected to germinate. And it's interesting that that's not really being affected by the treatments. And then in terms of germination success, so we ran some germination studies, and this table, it'll pop up in like a sequence, but we have ambient CO2 on the top, elevated CO2 on the bottom, and then their respective temperature treatments. So under ambient CO2, I just have the mean proportion germinated for each treatment. And you can tell that the highest proportion germinated is under 4.5 degrees above ambient, 4.5 degrees above ambient with ambient CO2. And under elevated CO2, the highest proportion germinated is when you don't add any additional temperature. So this is really interesting, given that seed mass wasn't being affected. So overall summary of findings to date, we have some preliminary data from the lab that show that at the highest temperature treatment, so at +9, trees are producing fewer cones, but they're bigger and contain more seeds with no treatment effects on seed size. We see some nonlinear responses in cone characteristics across treatments, with the full model explaining only cone dry mass and cone length, the temperature only model explaining the number of scales and presumably seeds if we had that full data, and the total and mean seed mass per cone not being affected by the treatments. So there's also differences in germination, as I mentioned. So under ambient CO2, it looks like when you add some temperature, you get increased productivity, so more cones, and germination is kind of higher in this like middle temperature. But however, because you have increased productivity, there are also changes in this carbon and nitrogen ratio because of these more cones, and that can have potential implications for seed quality and how well these seeds germinate and how viable they are. With elevated CO2, it looks like the addition of CO2 is beneficial if there's no temperature added. So we saw in the previous slide that germination was highest in the +0 ambient CO2 treatment, or +0 degrees Celsius elevated CO2 treatment. And yeah, so this can have potential implications on regeneration and forest composition in the future if warming and CO2 levels continue to rise. So I would like to thank my lab, the members of the Spruce team, as well as my advisors, the Oak Ridge National Laboratory, the U.S. Department of Energy, and the Natural Science Foundation for funding to do this research. And yeah, thank you, everyone, and I'll take any questions.
My name is Oscar Diaz. I am a master student at the Universidad Juárez of Southern Durango. And my team is the climate response of the genus Picea in northern Mexico. Okay. Yeah, the climate variation and change affect biodiversity ecosystems, ecosystem service, and the natural resource management. In Mexico, we have three populations of the genus Picea: Picea mexicana, Picea martinezi, and Picea chihuahuana. The population of Picea in Mexico are considered relict population and are in endangered status, given their restricted and limited location. Okay. Yeah, in Mexico, the climatological information is not extensive enough. Therefore, data obtained through road rains represent an important proxy for the reconstruction of climatic variables such as temperature and precipitation. Here, we can see two slides that were used for this study, and we can see how the climate impacts the population in different ways. The list of Picea martinezi, this left, and this is the list of Picea mexicana, right? Okay, therefore, the objective of this study is to analyze the annual growth of Picea mexicana and Picea martinezi population located in northern Mexico, and to determine which variable condition they wrote, right? Okay, five populations of the genus Picea distributed in different localities in the Sierra Madre Oriental were studied. And here is El Coahuilón, aquí es La Marta, here is El Bútano, here is Agua Fría, Afi, and here is Len. The red stars are the climate station. Yeah, the climate station was used to determine if there is an association with the climatic variables. Information was obtained from the meteorological stations closest to the sites. Yeah, in this graph, we can see the information mean of precipitation and temperature of any station. Okay, the methodology. From each population study, the individuals with the largest diameter, healthy and located in the sites of poor seasonal quality, for example, steep terrain, shallow soils, and poor fertility, were selected, where the select specimens depend exclusively on the rainwater for their growth, in order to generate the endocrinological series as extensive as possible, reduce the noise, the information rate to the limiting factor, and maximize the climate signal results. Yeah. Okay. Therefore, the suns were glued into the throats, yeah, like this, to be sent until the road rains could be clearly observed, like this. On this was done, a symbology based on points was signed. Each decade corresponds 1 point, like this. Each 50 years corresponds to points, and each century corresponds to 2 points, yeah? Then through an apparatus called Belmex, millimeter measurements were made of each round ring, taking into account early wood and late wood. This part is early wood, and this part in black, it's late wood, which together form a total ring, yeah? This is total ring. More examples, 1 point, 1 point, 1 point. Yeah, here's 2 points. Okay, ring with indices and several descriptive statistics we calculate. So ash are here is average radial growth, MS, mean sensitivity, first order autocorrelation, signal to noise ratio, inter-series correlation, and signal expressed for population. A principal component analysis was performed to determine if there is a common climatic variance and to work with the chronologies individual or jointly. Once the result was obtained, the ring width indices were associated with the climate data, taking a total period of 18 months, 6 months for the previous year and 12 for the current year. For this study, the previous months were from July to December, and the months of the current year were from January and December. Okay, the results. The statistical data, the directional seniors. We can see this statistical information. And a total of the 300 samples of the 5 population Picea were analyzed, having the longest series with the boot series, 275 years, and the shortest series are 144 years for the LMA and AFI. The Eco series has an extension of 230 years old, and the Lens series has an extension 148 years. We can see how the behavior over the last 14 years, approximately here, show very similar effects. Yeah. And with the growth well above average for the LM, LEN, and Eco populations. And to the illicit extent for the boom. In addition, all series show similarly low values for the recorded years in 1981, 1982, and 2011. The opposite shows that Picea mexicana population have a very similar behavior, so it was decided to work together with Eco and formed with Ecolma series. In turn, the population of Picea martinezi being geographically separated from each other and having very specific microclimates were worked separately. The first component explained of 59% and the second component 16.65%. This graph shows the correlation coefficients of the residual chronologies of Ecolma, boot, AFI, and LEN with monthly precipitation, monthly maximum temperature, and monthly minimum temperature. Conclusion, the results obtained from the diurnal series coincide with those previously reported for other conifer species in the region, which supports that the most important variable for the growth of the genus is the winter fertilization, January-May for Ecolma and boot, and January-June for LEN. The population of genus Picea, thriving in the Sierra Madre Oriental, shows a response function similar of that observed in the other space of the genus located at similar latitudes in the other parts of the world. These studies indicate the population of these species, Picea smithiana and Picea jesuensis, living at higher altitudes experience increased growth since 1980, where individuals shifted from the slow and steady increase to rapid increase in size, indicating that increased temperature simulated an air rate increase. The results of the present study suggest that the growth response of Picea populations to effects of climate change should be used in future models related to the dynamic of these populations. In addition, an aspect for the search future investigation is whether this growth response is similar to thought for other species associated with Picea in the Sierra Madre Oriental in areas with similar physiographic, altitudinal, and climate characteristics. Thank you.
Welcome to my presentation. My name is Florentin Jäger. I am a PhD student from Germany working with Christian Messier and Tanja Handa at the UQAM University in Montreal. And I am I want to present to you the results of my first chapter from my PhD thesis. And my first chapter is about the fine root biomass and distribution response to differences in growing season water availability of 6 different North American temperate tree species. And yeah, as you probably know, with climate change comes a higher intensity and duration and frequency of drought events, and also differences in precipitation regimes. And previous reviews and meta-analysis have found that trees tend to reduce their fine root biomass under a decrease of precipitation, and they increase biomass under increased precipitation. And they do that because under increased precipitation, higher precipitation levels, also they need more water to satisfy higher transpiration rates. And so for that, they need more fine roots, higher fine root biomass to satisfy this demand. And then under decreased precipitation, the decomposition of fine roots is higher, photosynthesis is lowered from the trees, carbohydrates that are stored in the roots are reduced, fine root turnover is irritated, and there is also a lower accumulation of fine roots because of a reduction in maintenance consumption for these fine roots. And yeah, I did not just wanted to look at fine root biomass and fine root biomass distribution in my first chapter, and that's why I also included the plasticity of fine root biomass and distribution in response to water availability. And what I found in the literature is that there is a tendency of faster growing trees to show a higher plasticity for fine root biomass in response to high and low water availability or changes in precipitation. And this is connected to the basic concept of early successional tree species versus late successional tree species, because early successionals have a higher biomass below and above ground at a young age, and they show higher transpiration rates and a faster root growth. So faster root growth, thinner and faster growing roots, fine roots, are presumably more plastic to changes, to environmental changes in the soil. And yeah, the late successionals, they show lower biomass below and above ground at a young age, and also slower initial growth, and then presumably lower plasticity in the fine root biomass in response to water availability. And yeah, the overarching goal of my study is to find more resilient, resistant tree species in the face of climate change, and to help to develop better eco-physiological models under changing precipitation regimes. And my hypotheses are that trees reduce absorptive fine root density under lower water availability, and that early successional tree species show a higher plasticity given their fine root biomass and distribution in the soil. And yeah, by itself, when you remember my first slide, this result that trees reduce fine root biomass under lower water availability, that's not new. But what is new here, that I could take advantage of this controlled experiment, the International Diversity Experiment Network with Trees site in Sault Ste. Marie. And there is a unique high and low water availability treatment installed. So half of these blocks receive a high water treatment and half receive a low water treatment. Those are 8 blocks in total. And I took measurements from 6 monoculture plots of 6 different tree species in each of these blocks. And the high water availability treatment means that these blocks and plots, they receive once a week during the growing season from June 1st to August 31st. They receive a high amount of additional water with water sprinklers. So that's about 130% above the average precipitation level in Sault Ste. Marie. And then the lower water availability treatment that consists of an annual removal of 25% of the naturally occurring rainfall there. And here you can see a picture with this quite unique gutter system that's installed there by the Ontario Forest Research Institute to remove this 25% of rainfall. Yeah, and so to assess plasticity, I calculated a plasticity index from Valladares, and I measured the root variables living and dead absorptive fine root density and mean rooting depth. And I did this by coring for the roots with this root corer here down to 30 centimeter soil depth. And then I took incremental measurements in 5 centimeter steps until 20 to 30 centimeter soil depth. So the last was 10 centimeter step. And yeah, here I show you my first result. So on the y-axis, you can see the absorptive fine root density. And then on the x-axis, you see the different soil depth layers for my 6 different North American temperate tree species. And you can see that there, for most of the species and most of the soil depth layers, there's not really a significant effect of the high and low water treatment here. You can see there is a bigger difference between high and low water in the upper soil depth layer, the 0 to 5 centimeter. And yeah, most of the trees show a tendency to increase the absorptive fine root density under high water here, especially in the first soil depth layer. And here I show you dead root density. And there again, I did not find a really a big effect of the high and low water treatment, but what we can see is a contrast between the conifers and the broadleaves. So here for Larix laricina, Pinus strobus, and Picea glauca, we see clearly that they have a higher deadroot density in the soil, and this is presumably because conifers tend to have thicker fine roots and more recalcitrant fine roots, and also they have a general tendency to put more roots down into the soil, more fine root biomass. And yeah, for the mean depth, only Larix laricina really showed a significant response to the low water treatment, but again, there is a general tendency for most of the tree species, except Betula papyrifera, to increase their mean rooting depth. And yeah, here I show you my plasticity results. So on the y-axis, you see the plasticity of the absorptive fine root biomass in the upper 5 centimeters of the soil over woody biomass grams per tree as a proxy for relative growth rate and successional status of my tree species. And we can see that the late successional tree species, Acer saccharum and Picea glauca, for example, that they show higher absorptive fine root biomass plasticity in the upper soil layer versus the early successional species, which here in this graph tend to show a higher plasticity for the mean rooting depth from the 0 to 30 centimeter soil depth. And in conclusion, I can say that the late successional species might be more vulnerable to future drought events or changes in precipitation regime because they are less able to plastically acclimate their mean rooting depth to these to the future climate change scenarios. And then to give a practical recommendation, perhaps it's advised to, from my plasticity results, of course, to plant more larix and early successional broadleaf species. But of course, I have to mention here, I can only make this statement in response to water stress. I know there are many more stress factors. And yeah, then for the fine root biomass response, the first graph that I showed, it really seems that under high water, the tree species, they want to maximize the exploitation capacity in the soil by investing much more into their fine root biomass. And then under low water, they try to reduce risk of fine root loss through desiccation. They put less fine root biomass into the soil. And yeah, the increased rooting depth, the increased mean rooting depth of Larix laricina under low water, that could partly compensate for the reduced water uptake that is happening presumably because of lower absorptive fine root density in the upper soil layer under low water. And finally, I want to thank my collaborators and my assistants, most notably to Dr. Pedro Antunes from the Algoma University and Akihiro Koyama from the Algoma University that have helped me a lot in the field and the lab with collecting the root samples, washing the roots, scanning the roots, the many steps that are necessary, and Isabelle Aubin, who helped me with facilities and technicians and material. And then, of course, my colleagues from Tanja Handa's lab and the lab of Christian Messier. So thank you very much for listening, and I'm glad to hear questions, to receive questions.
I'm going to give you a little bit about background of the area, what we did, why we did it, and then some of the outcomes. So Northeast BC is mostly complex conifer broadleaf forest, and in BC, the regulations, basically based on economics, push management towards even age conifer stands. And the goal is to maximize conifer production. And so because the economic value is not there in broadleafs in the northeast of the province at this point in time. So the goal, we're maximizing conifer production, but not site productivity. And all of the standards for management are based on competition metrics and not tree growth. So the typical management pattern is shortly after establishment, you remove the broadleaves. And this results in a management contradiction because we're supposed to maintain what we saw at the landscape level. However, it doesn't really appear to work that way. So we reduced diversity, both structural and species. And in an earlier study we looked at the economics of this broadcast brushing, and it doesn't really make sense economically, even as well as biologically. So the climate in Northeast BC is projected to increase by 2050. So the mean annual temperature is going to go up roughly 2 to 5 degrees, depending on where you are. And the 3 sites that I've indicated are pretty close to where the sites are that we're going to study. And you can see that we have quite a temperature extreme even now in the region. Precipitation is going to increase roughly 10 to 20% on a 450 millimeter annual. And frost-free days are going to increase by roughly a month from the current 3 to 4 months. So really big changes for the region. And in BC, we have a facilitated migration program. But in Northeast BC, we have very heavy soils, and that would be suitable for the new climate are not suitable for the soil type. So anyhow, another question yet to be answered. So we have little or no experience in the Northeast with complex stand management. And by doing what we're doing, we're essentially losing the non-timber benefits that we get. So we're losing biodiversity and resilience. First Nations are not happy with the way the land is managed. We're losing habitat value, and we lose pest and insect resistance. So a lot of negatives to the way that we are doing our current management approach. So our objectives were to look at the range of stands across the Northeast that hadn't been brushed. And we have species mixtures. We have aspen, paper birch in combination with white spruce in various sites. And our underlying assumption was that interspecific competition was less than intra-specific competition. And this is just based on our experience in walking through these stands. So the 3 stands, one is south of Dawson Creek, and it was 18 years old when we established it. One was just north of Fort St. John, and it was 5 years old. And the other was south of Fort Nelson, and it was about 14 years old at establishment. We used Picea, which is a growth and yield model, managed stand tables, and it's the benchmark for timber supply analysis in BC. So we were going to see the growth that we got versus what was projected in this model. And the inputs for the model are relatively straightforward, activity and whatever treatments you're providing. So at establishment, this is diameter of the trees that were there versus stand density on the x-axis. And I guess somewhat to our surprise that there really wasn't major fall-offs or abrupt fall-offs in diameter at establishment with competition, levels of broadleaf competition. And then when we looked at growth, we got, I think, patterns that were a little more to what we would expect, in that at low densities you have better growth than at higher densities. But the question still exists, is how acceptable is the growth that we're getting with no treatment, which is what we're looking at here. So then we ran the model, and so the spruce bars are what we actually saw from the growth, and the under the site indexes are the 3 sites. So the blue is site 1, the orange is site 2, and the gray is site 3. And you can see that our diameter growth at the sites is as good or better than at the model projections. And when we look at basal area, which is a surrogate for site productivity. Spruce basal area is pretty much as expected at site 1, so the competition hasn't impacted, and the spruce basal area is much better at sites 2 and 3. So we haven't apparently impacted the productivity of the spruce, but the total basal area for the sites are significantly greater than and have enhanced productivity on the sites. So stand density affected crop diameter, but nothing very exciting, I think you would say. And the stand basal area measured was at least twice what was projected in Picea. So this suggests that mixedwood productivity is greater than a pure spruce stand, and we're having minimal impact on tree size. So the pictures that I put in here are really to give you an idea that this really is a mixed forest. We don't have the only large patches of pure green that you will see are the plantations that have been managed and the broadleafs removed, but mostly it's a heterogeneous landscape. So we do have competition thresholds for sure. They're high by current thinking, but they're in the 4,000 to 10,000 stems per hectare of broadleaf range. So is that important? It's hard to say. Mean spruce diameter was not really impacted, and this is across all sites. So that's a good news story. Spruce height exceeded projections on 2 of the sites, meaning that 2 things. Maybe the productivity of the sites was underestimated from the methods that we used, and it was about what was expected on the 3rd site. Stand basal area was much greater than the Picea spruce basal area, so we have not impacted overall productivity. And the spruce basal area was greater than what was projected in Picea. So not only have we not impacted, we've got more productivity, but we haven't impacted the spruce productivity. So we're trying to manage this on a very heterogeneous landscape. We don't have a lot of experience. So how are we going to manage to do this? And that's really a big question. We need to understand the dynamics of complex stands, and people in Scandinavia are having similar issues. That what is the minimum we need to do to maintain the complexity? The total productivity is greater in the complex stand, so this is certainly encouraging from many points of view. But this does not or will not apply or work everywhere in the Northeast, because not every stand has the attributes of the ones that we were directed to by colleagues in the area. So Northeast British Columbia mixed woods in a changing climate. Broadcast brushing may not be a good investment, both from an ecological and a financial point of view. We get a greater total yield of carbon than the pure spruce of the broadleaf stand alone. Conversion to pure conifer stand reduces diversity, species structural on both at stand and landscape levels. And it does not meet First Nations values and land use objectives. And this is very important because in the Northeast, there's just been a recent court decision about essentially both forestry and oil and gas negatively impacting on First Nations traditional territories and on their traditional land use objectives. So I wouldn't say it's a hiatus, but Going forward, land use is going to be very different, I think, in the Northeast than it has been. And managing
Well, good afternoon, everyone. My name is Erin Pearson. I'm a Master of Science candidate at Memorial University, and today I'm going to be sharing my master's research, which is looking at urban forests on the island of Newfoundland. So urban forests provide a variety of benefits and services to the urban ecosystem, including the mitigation of climate change impacts. However, in the face of ongoing climate change, urban forests must now adapt to new and changing climate conditions. And the ability to adapt varies for a number of reasons, including its ecological integrity. Additionally, urban forest ecosystem services are maximized with increased levels of ecological integrity. Therefore, as environmental and climate conditions are changing, the ecological integrity of the urban forest plays a crucial role in both maintaining the urban forest and mitigating climate change impacts. And the island of Newfoundland is an ideal location to study the relationship between urban forests and the urban landscape, because, as I will explain in just a moment, it may reflect the future of Canadian winter climates. So we developed two main research objectives for this project. Firstly, to determine how local climates influence the ecological integrity of urban forests. And secondly, to determine the extent to which local climate change impacts are mitigated by urban trees. So our approach to addressing these objectives was to implement two different gradient analyses. And these influenced our decisions in choosing field sites, which are 4 urban centers on the island of Newfoundland. So we have Corner Brook, Gander, Conception Bay South, and St. John's. So the first gradient that's represented here is related to winter climate regimes, specifically temperatures and wind trends. So Corner Brook on the west coast tends to experience reliably cold winters with relatively low wind speeds and infrequent severe wind events, while St. John's on the east coast experiences milder winters with more frequent high. I mentioned this reflects the predicted change in Canadian winter climates. So currently, winters in Canadian cities are reflective of the West Coast. So they generally have cold temperatures with lower wind speeds. And this is shifting as a result of climate change towards a winter climate more representative of the East Coast. Temperatures are predicted to increase along with wind speeds and severe wind events. Therefore, the island of Newfoundland can act as a worst case scenario model of the impacts from rapid warming and increased winds on existing tree species. So then we have the urban forest gradient. So first here we have peri-urban areas, which are large areas of natural forest within or nearby the urban center. Then we have treed urban areas and untreed urban areas. So there are many different ways to measure ecological integrity. My focus was on factors that influence the substrate, community composition, and structure. So 10 plots were established at each site type with a central tree or utility pole acting as the center of the plot. First, we used a 1 by 1 meter plot, and here we recorded the ground substrate cover and soil organic matter depth. I also conducted a ground vegetation survey and recorded the canopy cover, canopy height, total tree height, diameter, and species identification of that central tree. A larger 5 meter radius plot was then used to measure the species composing the immediate surrounding of the tree, their diameter, and their position in the canopy. So these measures help us assess the ecological integrity of each plot and then compare across our 2 gradients. To display our ecological integrity results, we performed non-metric multidimensional scaling. So this technique ranks sites according to their similarity. So those that are closest together are most similar in their ground cover types, and those farthest apart are least similar. So this graph is going to display all of our sites, The colors represent different site types. So green is our peri-urban, pink is our treed urban, and gold is our untreed urban. And the shapes represent the different cities. So when we add our sites to the graph, we can see that they're generally grouped according to site type. So the peri-urban in green are most similar to other peri-urban sites, but are least similar to the untreed urban sites, which are in gold at the other side of the graph. We can also add ellipses here based on the site types that represent the 95% confidence interval, meaning that 95% of the variation in the site falls within the ellipses. So here we can see that individual sites of treed urban areas have a much higher variation because the ellipses is much larger than it was in peri-urban and untreed urban. So based on this information, we can see that peri-urban sites, even in different cities, have very similar ground cover types. This is also true for untreed urban sites, and peri-urban and untreed urban are not that similar to each other. The treed urban kind of acts then as a gradient between the two different sites. Some are more like our untreed and some are more like our peri-urban areas. So here we can now look at what's actually on the ground. When we add the actual ground covers, we can see that peri-urban areas are mostly composed of more natural surfaces. So things like bare ground, moss, rocks, and coarse woody debris, while our untreed urban sites were mostly composed of the utility pole, grass with some pavement. And the treed urban kind of have elements of both. So what does this mean? The untreed urban sites are generally composed of less permeable material. So when we have a lot of precipitation, we can get large scale flooding in untreed urban areas. This also then means that there's less nutrients available for trees in untreed urban areas because all of these really great natural surfaces like coarse woody debris and moss provide nutrients to the trees, and they're not as present in our urban environments. We can also look at the relationship between ground covers and different environmental factors, including different vegetation groups. So these vectors represent different vegetation categories, and the length and direction of the vector indicates the strength of that relationship. So for example, the Conifers vector way out here is quite long and in the direction of our peri-urban, meaning that there's a relationship between peri-urban sites and the presence of conifers. So as we move from untreed urban areas into more natural peri-urban areas, we'll start seeing more conifers. Two other important aspects of ecological integrity are also present here: species richness and native species. Both are increasing in peri-urban sites, meaning that as we transition from peri-urban into treed urban and then untreed, the overall species diversity and abundance of native species is decreasing and non-natives are increasing. So these indicators suggest that ecological integrity is highest in our peri-urban areas and lowers as we move into increasingly urban environments. To determine the extent to which temperature-related climate change impacts are mitigated, we installed an above ground and below ground logger at each plot. So here I'm going to show you just the results of mean temperatures from Gander as an example, starting with below ground. Here we can see that in the winter months, the temperature in peri-urban environments is higher than in treed urban and untreed urban. And then this switches in the spring and summer, where peri-urban is the coolest of all three site types. So this is evidence of the buffering effect that's provided by forests. They create an insulating effect in the winter months, keeping temperatures relatively mild, and they reduce the amount of solar radiation reaching the ground in warmer months, lowering ground temperatures. So then we can look at above ground, and the first notable difference here is that there's much higher fluctuation in above ground than there was in below ground. So a lot more peaks and valleys in the graph. Secondly, peri-urban generally remains cooler than the other site types, but the difference is not as obvious. This is likely due to the effect that wind has. So even in Gander and Corner Brook are areas that had relatively low wind. Wind is still a really big factor in our local climate here in Newfoundland. And this reduces the ability of trees to create that insulating effect, reducing the impact they have on ameliorating temperatures. I also want to really briefly show the temperature trends for Conception Bay South. So since the differences in temperature at each site are a bit more extreme here, we can also see another interesting trend, and that is the difference in variation around the mean. So if we look at the end of April through September, the variation in temperatures evidenced by the translucent ribbon around the trend line, it's highest in untreed urban areas and lowest in our peri-urban areas, demonstrating the ability of trees to ameliorate daily temperature extremes. The last of our climate mitigation is still underway, and these analyses generally fall into two different categories. We're looking at daily temperatures, so how daily temperatures differ along that urban forest gradient, and weather and climate events, so how the occurrence and duration of these events like frost and freeze thaw and heat waves differ along that urban forest gradient. The expansion of the urban forest is a major component of climate mitigation and adaptation strategies for municipalities, yet a clear understanding of the benefits they provide and impacts to the urban forest are needed. Urban centers in Newfoundland are somewhat unique compared to other regions in Canada and North America, given that they are smaller in size and our climate is generally relatively regulated by the Gulf Stream and Labrador Current. Therefore, there's minimal urban heat island effect when compared to larger urban centers located further inland. Yet we still see here that there's a clear benefit from the urban forest in terms of temperature mitigation, and we see that the urban landscape has a clear impact on the ecological integrity of urban forests, meaning this is likely more pronounced in larger urban centers and will become more prominent in Newfoundland and other larger urban centers as climate conditions are changing. So I want to thank you all for listening, and thank you to everyone who's helped me get to this point of my degree, including my incredible supervisors and lovely field assistants. And I'll now open the floor for any questions.